Fantastic.
Okay. Okay, so welcome all to the, I think this is the third talk in the Turing series. We'll be having a couple more talks. And so today I'm going to be talking on Turing's work on mathematical biology. So Turing was definitely a versatile man and did a lot of different things. And so the title of the talk is, Why Do Tigers Have Stripes? And so this was one of the central questions he had. Why do these patterns involve? And so the overview here is, first we sort of try to figure out what motivated Turing. Then we'll get into his actual work on morphogenesis. And then the impact of the work, how this motivated others, and his legacy. So Alan Turing had three seminal papers, basically, which have had over 5,000 citations each. And before this talk, I was quite familiar with two of them. So the first one is on Turing machines, where they introduced the Turing machine. The second one is the Turing test he designed for artificial intelligence. So next week, there will be a talk on Turing machines. And next month, there's going to be a talk on the Turing test. Now, before a few weeks ago, I wasn't familiar with the third one, which is his work on pattern formation. But this is definitely an interesting paper, and it has received over 5,000 citations, which is quite a lot, and more than most people can expect for all their papers produced in lifetime. So, and it has definitely had a great impact. And so the question he was asking or thinking about is, well, basically this. So life of an organism, it starts from a single cell. This cell is completely patternless. So there's nothing there that, if you look at it in a microscope, and if you look at maybe the different embryos from different species, there's no recognizable pattern in it. There's a nucleus maybe, and a couple of other things, but there's no pattern there which you can see. Yet it evolves to something that's very complicated, and sometimes it has these very complex patterns, and the pattern is often the same across species, and usually the same across species. So, the Turing's thinking, or my interpretation of his thinking, is that there must be something that tells the species that you should be zebra. Well, you're a zebra, so you have to have stripes, and you're a leopard, I guess. And then you should have these sort of patterns. And something that tells them, over and over again, so why don't we have any zebras with these dots? And why aren't there any leopards with these stripes? I mean, so it's a separate question from why are zebras black and white? But, so these were the questions that were motivating them. And these actually have been questions that have come up in the centuries. And so Turing worked on this from 1952 until his death in 1954. Even though he was working on a couple other projects, this was sort of his main intellectual challenge. So he published this paper in 1952 and put forth this Turing hypothesis on pattern formation. Now, so the pattern formation question has come up through the centuries, and in Western society, this was first suggested by Fibonacci in 1200, where he gave his famous Fibonacci sequence, where he showed that this sequence occurs many times in wildlife and in different species. Now, so basically, the morphogenesis is what we're going to be talking about, which is the study of shapes of tissues and the positions of various cells. So this was maybe the primary motivation for Turing's work were these Fibonacci patterns. And so you have this sequence here so the Fibonacci sequence is basically you start with 1 and 1, and then you make the next term by summing up the two adjacent terms. So you get 1, 1, 2, 3, 5, 8, etc. And so you can see how this occurs here in like an idealized version of a flower tree. So basically there has one stem here, and again one stem which splits up to 2, then split up to 3, 5, etc. And you can sort of see the same pattern here in the nautilus, how these sort of rings keep growing each time, and the growth of these rings is basically in this sequence. So here you have the sunflower, and you have a similar sort of progression of this pattern where you have ever-growing things out of the middle of the sunflower. Now, okay, so now that was the motivation. So now let's get into the theory of this. And so at this time, when Turing was doing this work, biology was far less advanced than currently. And for instance, the structure of the DNA wasn't published till after Turing's death. So there are a lot of things that people didn't really understand or know about. But Turing defined these entities called morphogens. So he said, okay, so there's something that he's going to call a morphogen, which we say is a substance that governs the pattern of tissue development. And it spreads from some source, and it forms some sort of concentration gradient across the different tissues. And so if you have a lot of this morphogen, then you get a specific picture. So basically, you can think of this as, the morphogen here is occurring in each one of these semi-rings here. Each place where there's a semi-ring, there was the occurrence of a morphogen. And so in development biology currently, these morphogens are thought of as signaling molecules, and that have some sort of effect on other molecules. Now, so this was his description, the description that Turing gave for modeling embryogenesis, so basically the development of an embryo. So he says, okay, so the model is of course a simplification and an idealization, and so it says consequently a falsification, and so he doesn't really, he tries his best to not define what a morphogen is. And it just says it's some kind of a substance in this theory. So because there's no real knowledge in this in biology, And I mean, part of the paper is that he develops a lot of theory, but he's quite aware of that fact that there's very little in terms of experiment that he could actually do, or anyone can do at that time. And so it's some kind of substance, and it diffuses into the tissue, and somehow it persuades it to develop along different lines from those which would have been followed in its absence. So, you know, in the 21st century, we know of plenty of these different morphogens. But at that point, he didn't know of any. So diffusion is the standard model people had for movement of molecules. So you've probably seen this again, seen this before. You put a few molecules in a bag somehow, in a large bag, and what happens is that they spread out all over the bag and become somehow sort of evenly distributed, and they don't leave these vacuums. So now this clearly was a sufficient model for the single cell embryo. But it wasn't a good model for a complex organism because, I mean, unless you're looking at an amoeba, I mean, the organism isn't all just the same single blob. There are definitely different things that are forming there. And so, yes. So one of the key observations is that also that we know now, at least, is that these are forming all from the same recipe book or the same DNA. So there's the same program that actually gives these, but the morphogens start the program from different places. So the paper actually is quite long. It's 37 pages. And it's quite an involved paper because it relies on fairly complex ideas in chemistry, biology, and mathematics, and a little bit of computer science. And so here's my basic attempt to explain chemistry to you. So there's no real chemistry needed for this, and I definitely am not a specialist in chemistry. But so you have a reaction. So here we have a picture. So here you have two sets of molecules. So there are two chlorine atoms attached together and two hydrogen atoms attached together. And you mix them together, and what you get is that there's a reaction between these things, and they create hydrogen chloride. And so the rate of this reaction is dependent on how much you have of A and B. And in this talk, we're going to use A both to represent the chemical and the concentration of that chemical. So we'll use A here for the chemical A, or hydrogen, and the concentration of hydrogen. So how much hydrogen we actually have. So basically, if we have a lot of hydrogen and a lot of chloride, then this is going to go quicker than if we have little. And so then we're going to build a model for where we have multiple cells, and we're going to think of each one of these cells as a unit. And the basic thing with diffusion is, even though you have these cell walls, the idea is that these morphogens, they're small enough that they can actually permeate these cell walls. And so basically, if you have a lot of one morphogen in this cell, then in the adjacent cell, you should have a lot of the morphogen as well. And so the theory would then say, then if you have the same amount of the morphogens in the cells are going to look similar, and we're going to get a similar pattern, which again would then mean that all the cells would be the same. Okay, so he says, I mean, in this paper that, okay, there is a steady state that the concentration of these morphogens reach in the different cells, but it's different between the cells. So it's spatially homogeneous. But there's a clear reason why there could be a pattern actually forming. And so this was the fundamental insight as people thought that diffusion would basically stabilize the concentration of all the different cells. So here is the model that you have. Suppose we have two morphogens, and let's call them x and y. And remember, we're going to use x just for the morphogen and the concentration of that morphogen. Then suppose we have these two morphogens, then each one of them is going to be produced via some complex chemistry at some rate, and we can assume that this is dependent on x and y. So there's one function for x on the production of x, and there's another function for y on the production of y. Now, okay, so let's look at this simple example here. Suppose that x is produced at the rate 5x minus 6y plus 1. And y is produced at the rate 6x minus 7y plus 1. Then if we put x equal 1 in here and y equal 1, then we see this is 5 minus 6 plus 1, which is 0, and 6 minus 7 plus 1, which is 0 again. So what we have is that there's an equilibrium. There's no production of x, and there's no production of y. So we can get an equilibrium. But what happens if we increase x just ever so slightly? So if x increases and y stays the same, that means that this goes just ever so slightly above 1. So we start producing x. And we actually start producing y as well. And, well, so if we start producing x, that also means that there's going to be more x. So we start producing more of x. And there's going to be more of y. So we get this chain reaction where we produce ever-increasing amounts of x and y. And so this was one of his fundamental observations in this paper is that, okay, so you can have an equilibrium here, but it's a very unstable equilibrium. So basically, if either x or y moves ever so slightly from 1, then both x and y are going to get produced or removed at a rapid rate. So, and the thing is, if you have diffusion, then there's always going to be a small fluctuation of the concentration of x and y. So the diffusion basically ensures then we cannot have these unstable equilibria. And so we move from it. So okay, so he describes a set of reactions that can describe the above formulas. It's actually, the numbers here are a little bit artificial. 5x minus 6y plus 1, 6x minus 7y plus 1. But, he says how we can actually produce these things at that exact rate. So you need a set of reactions producing the first morphogen at a constant rate 1. And also for the second morphogen, you need a set destroying the second morphogen at rate 7y, a set converting the first morphogen into the second at a rate 6x, and a set producing the first morphogen at rate 11x. And then you need a set destroying the first morphogen x at rate 7y, as long as any of it's present. So basically, you need 5 different reactions to getting that set of equations up there. And okay, so in general, what you can do is you can set up these differential equations. So what we're going to assume here is that we've labeled all our cells here from 1 up to n, and we can assume that they're cyclic. So they're occurring in a cycle, so it's a ring. So it's the tummy of the zebra. And so generally what it says is, okay, so the change in the concentration of x is some function of the concentration of x and y plus some function of the difference in concentration of x with its adjacent cells. Because if there's a difference of concentration in the adjacent cells, then there's going to be flow between the cells. This is the diffusion part, and this is the reaction part. And the similar for YR is that we have a diffusion part here. We have a reaction part that Y is going to be created or destroyed, and that is some function of the concentration of X and Y. And then it's going to diffuse from its adjacent cells, and that depends on the difference in concentration with its adjacent cells. And then he shows, okay, we can actually make this system a linear system, making some assumptions, so that the change in concentration of X is only dependent on some some constant times the concentration of X plus some constant times the concentration of Y, and plus this diffusion term. And so these were basically his formulas. And you can actually put this in a fancier notation now using contemporary mathematics, if you like, and you get the similar sort of idea. Yes. So basically, his contribution was that you may have these reaction equations, and you may actually have an equilibrium to these equations, but if you add in these diffusion parts, then that equilibrium will not hold, and you move away from the equilibrium. And so instead of these solutions where the concentration is equal between the different cells, you actually get the solutions where the concentration may actually oscillate between the different cells. And So basically, the main or some of the contributions to the paper were that he noticed that these differential equations will be unstable in the presence of diffusion, and he characterized the solutions of these equations, and then he mapped the solutions of these equations to different biological events. And so here are sort of some of the different forms that these solutions may take, and you actually can see that you can get these wave patterns. So this corresponds to the zebra patterns, where one cell has high concentration of the black morphogen. The adjacent cell has slightly less until you reach a cell which has no concentration of black creating morphogen. So this would correspond almost exactly to the pattern of the zebras. And you can have these exponential growing functions, or you can have these growing waves, and you can have a few other different things. And he actually said, sometimes these solutions, they're going to be very stable, and sometimes they will be less stable, which means that, like in a nautilus, there's a high amount of structure in the difference between the size of the rings, but in zebra, there is a greater difference in the stripes between the different zebras. Okay, so here are some pictures of how these solutions actually how these morphogens can actually arise, and you can see that these corresponds to different patterns. And so you can note these things that if you have so it depends on the domain of the solution space, what the different pattern is going to be. So basically, depending on how the organism is organized, there's going to be different solutions to these equations. So this was Alan Turing's observations of his theory. He says that my theory of embryology is yielding to treatment, and as far as I could see, gives satisfactory explanations of gastrulation, so which is the formation of a gut, and polygonally symmetrical structures such as starfish or flowers. So here starfish, basically you have one of these solutions to equations will form a starfish. Leaf arrangements, in particular the way Fibonacci series come to be involved, as these, for instance, and color patterns on some animals such as stripes, spots, and dappling. Okay, so that was basically the overview of his results, and so we move to the second or third part of the talk, which is basically some further observations of his paper. So his main observation here was taking a system that without diffusion is stable, but adding diffusion is actually going to be unstable. And he managed to show that you can actually turn this into a linear set of equations, which means that this also computationally feasible to work with these equations. So a couple of other observations people have made about his paper is that he did make the observation that as opposed to most physical laws known at that time, biological systems did not have these perfect mirror image symmetries, or not the type of symmetries that physical laws had. Now, and so another observation people made is that this paper marked the transition point from analytical mathematics to that of computational mathematics. And so, I mean, one of the motivations is that Turing at this time was involved with building a computer, and he was trying to find use for this computer. And so he was trying to motivate people how they could actually start using computers to do interesting science. And or you know, he could, maybe was one of the first people to see that this was a possibility. Now, so I said that this paper had over 5,000 citations, and so I made an interesting observation here, which I thought was interesting. So, which are the 10 papers that are most highly cited that cite this paper? So, you know, we'll look at the 10 most important papers citing this paper. And so most of the time, you know, you write a paper and it's on differential equations. People that solve differential equations, they will read the paper and they will cite your paper. This is not the case for this paper, and it actually one of the reasons is that it involves mathematics, it involves chemistry, and involves biology. But it actually got cited by many different other different fields. So the top citation here is, the most highly cited paper cited in this paper is a book on mathematical biology, which we would sort of expect. The next is a paper on directed diffusion for wireless sensor networking. So this is, people are trying to put sensor networks around the world, distribute them in a sensible way. The third paper is by our Nobel laureate Krugman, spatial economy, cities, region, and international trade. And there's a paper here on synergenetics, which is another field of biology, and pattern formation in physics, and pattern formation in chemistry and ecology. There's a book here on reflections on genders and science, and I didn't really try to understand how that was related to this paper. And there's another paper here on sensor networks, and there's a paper or book on differential equations. Now, okay, so Turing's, one of the reasons I got involved in this was also just to figure out Turing's impact in computational biology and sort of figure out, okay, so what you know has this paper actually taught us? I mean, some papers from the early or 1950s are still maybe representative, but I mean, a paper in biology that from 1950s is really different than current biology papers, because there's been a lot of different progress in biology in that time. So, so Turing's impact in computational biology is, so his main impact, I think, is suggesting basically that life could be considered as a computational machine. So, you know, one of the things he was trying to say there in this paper is that there must be some program that tells the organism that it should actually develop in this way. And, you know, what he was sort of his main question was in his life really, what is computational? What can you reduce to computation? And he was maybe trying to say that life could be considered as a computational machine. And so he definitely had a lot of impact with his Turing machine and the Turing test. And those have definitely given people a lot of insights into studying computational biology. So this is now currently, this was not known at that time, the central dogma for biology is that basically you have DNA, which you can transcribe to RNA, and which is then translated to protein. And the idea is that proteins, they actually do most of the work inside the cells. And while the DNA here is basically what you can think of as a hard drive, which stores all the information about how the different cells should work. And the RNA is basically a copy here, a short copy of the DNA that later gets translated. And so the morphogens, they're not really working on the DNA at all, so they're not changing the DNA per se. They're changing what gets transcribed from the DNA. They're changing the structure of these proteins and how they interact with each other. Yeah, so that's the same picture again. Now, so the other contribution, or one more contribution here, is the Turing and von Neumann machines. So Turing, prior to this, had invented the stored program computer, and yes. So von Neumann showed that this description was separate from the universal constructor, and at that time, people had not realized this separation between the DNA and the proteins and the RNA. And so even Schrödinger here wrote in his book, What is Life, where he saw that chromosomes were the architect's plan and builder's craft in one, while the Turing was making a different point here. And so von Neumann suggested here that we should try to construct a machine that could replicate itself, and this was very much motivated by Turing's work. And we can, currently people view biology as an example of these Turing and von Neumann machines. Okay, so to further on that, we can view animals as some highly complex programs that are regulated by these different gene expressions or morphogens. And these are regulated by some network of interactions of proteins and chemicals in the cells. And currently people are working on a lot of these different type of networks and sort of trying to figure out how actually all the different components here interact. And so basically the challenge is, okay, it's currently accepted that there is a program that regulates each organism, but people are trying to figure out, okay, so what is this program and how actually does it work? And so people have more complex vision of what actually is a morphogen, and there are different things that actually are morphogens. Okay, so Turing at this point of publication, we should note that he was actually quite a celebrated scientist at the time. He was 38 years old, I think, and he was recently he got elected as a Royal Fellow of the Royal Society, which is one of the most prestigious things you can get in British academia, at that time at least, and basically meant that you were a senior professor. And this was something that people that were 38 usually were not awarded. I'm 38 and have not been appointed a Fellow of the Royal Society yet. But he was in charge of building the first computer, and not the first computer, but one of the first computers in Manchester. So there was a big, actually, a big building and laboratory for building these computers. And so this was a lot of technical work, which he got highly involved with, but this involved a lot of managerial stuff. So one of his design was that he used 32 bit words for his computer, and because they didn't have any programming languages at the time, he had to have a very complex arithmetic for doing arithmetic in 32 bits, which was very hard for most people to understand, but apparently quite easy for Alan Turing to understand. So he seemed to get frustrated by people that would think his 32-bit notation was cumbersome, and multiplication in these 32 character words were cumbersome. So and somehow computers were not thought at the time as real science, or a lot of people sort of frowned on computers. It was basically a big waste of money, and you are computing something you could already compute anyway. So why do these things? And it wasn't. It was more of it wasn't the thing that a Fellow of the Royal Society should be doing. But he liked to work on pure science, and so that's one of the reasons he got into this. And so, yes. So here is sort of his philosophy or interpretation of his philosophy, and for Neumann, which was a collaborator of his. So, what was his work ethic? And basically, so, be an automata theorist. So basically, he was working on these automata things. But the work ethics are basically work on the theoretical and the practical problems at the same time. So he made sure that he was working on theoretical problems at the same time as he was working on practical problems. And use any method that he can use. So be a mathematician of the discrete and the continuous. And be intramath, inner science, a cross-culture scientist. So he actually, in this paper, he was venturing into biology and into chemistry and mathematics. And make sure that you're working on the hardest problems. And make sure you love what you're doing. So what followed this publication is that soon after this he actually got involved in a relationship, and then he was arrested and didn't do very much after that. Okay, so that's a talk. thank you for listening.
So we have time for questions.
What is Leonardo DiCaprio doing there?
Oh, so actually there was this book about Alan Turing, and Warner's Brothers bought the right for the books, and he's going to be playing Alan Turing in a couple of years. So this is the future. You can wait to see the film, since you've already seen the talk, you can see the film. Or you can buy the book. The book is pretty good, but it's long and it takes a long time to read these things. Look at the end of the book, and I only managed to read 20 pages of it.
So, Bjarni, I have a question.
Yeah.
So, what do you think has been the impact of Turing's paper on mathematical biology? I think sort of recently, very recently, I think there was a paper in Nature Genetics which seems to vindicate his theory, and it had to do with tigers.
Yeah. So, I mean, it is quite of a simplification. So, whenever you have a paper that is such a simplification, I mean, it's really open to a lot of interpretations of what the impact is. But I mean, it definitely has been cited by 5,700 publications. So the impact is not only just in this pattern formation. I mean, it motivated a lot of people to study pattern formation. And I mean, some of these equations, they work for some things, but they don't work really for everything that Turing suggested they would work for. So, it has had a lot of impact, but I mean, the paper was a bit more far-reaching than it actually could be, because it was so far away from the data measurements. But yes, people have actually seen a lot of these patterns, and I mean, you do have these measurements from Fibonacci in the 12th century, where you actually see these patterns. So
I think also, as you mentioned, he was, I think if you take the introduction, the beginning of the introduction to this paper, it's one of the most honest introductions you can ever find in science. He basically says, here's my model, it's a simplification, it's a falsification, but I still hope that what is retained is of interest, and what the conclusions that I can draw from it are reasonable. And I think that's, today you will not get that many papers accepted if you start.
Where did you publish this? Like what type of venue?
Yeah, so that's a good question. We can use our friend Google here. I should know this, but I'm thinking it was Proceedings of the Royal Society. Okay.
You can try Wikipedia, I'm sure.
Here's the paper. Yes, Transactions of the Royal Society. Series B, Biological Sciences. So it made biology. So it's basically, I mean, it's a biology paper more than anything. But I guess since he just got the FRS, he could publish there.
Yes, you can also see the beginning of the introduction. Yeah.
Yes. Yeah, I mean, so there's a good reason why that's a simplification, and I mean, because there are no measurements. So he's developing a lot of theory. I mean, I think his greatest impact is really starting to think of an organism as a computational machine, and there's a set of computations, and in that sense, he has had a tremendous impact, and everybody now thinks of of these things as computational machines. And if you used Schrödinger's idea, I mean, I don't think that would have worked. So there were definitely people that wouldn't agree with his model at that time.
Okay, so if there are no further questions, we thank Bjarni again.
Yeah.
Oh, yeah, there is a question. Sorry.
Is there any work on in this line here at the university, pattern recognition, biological pattern recognition? Is there anyone working on this issue?
Not pattern recognition per se, but I've worked a lot on computational biology and they sort of trying to discover these computational machines, but not on the pattern recognition, no.
I'm just trying to.
Yeah.